Again, I enjoyed the conference, and this has been fantastic. Sorry about the problems that occurred right after I was raving about what a great conference this was. But let me just say my company is Sierra Sciences. My website is sierrasciences.com. And I want everybody to know that because that's the best way to learn about what we're doing because I can't possibly say everything in 30 minutes. But our goal is to find a cure for aging. And this is what I mean by aging. I'm not trying to, I would not be satisfied by just reversing some biomarkers, even though I consider reversing biomarkers very important. This is what I'm after. I want to actually see somebody like Betty White go from, in this picture, she's 95, she's really 99 now. I want to go back to seeing her like when she was 25. This is what my mission is. I call this the Betty White test. And as far as I'm concerned, aging has not been cured until people start passing this test, not just a bunch of biomarkers. But a main reason why I think it's important to cure aging is because a lot of great things are going to happen in the future. And I want to be there for all of those. When we meet intelligence, life on other planets, I want to be there. This is why I am so focused and so obsessed on trying to find a cure for aging. I want to spend a few minutes on who I am. I'm going to tell you that I'm going to go fast through most of the slides. This isn't not going to really be a science presentation today. I'm going to be talking about more other things, but I want to just give you a description of who I am and how to learn more about me. But I am a medical researcher. I do have a PhD in molecular genetics and evolution. It's also called population biology. I've been in biotech for 40 years. Nuno did say I got my PhD in 1991. I actually got it in 1981. So I'm actually in the field for a lot longer than that. I was actually, as Nuno mentioned, second place for National Inventor of the Year for my cancer research. I don't just do aging research. I've got a big background in studying all kinds of diseases. I'm also an ultramarathon runner. I believe in making athletics important, and I've been on 2 documentaries. One Nuno mentioned called The Immortalist. I've also been in a documentary called The High. The High is more about my running, but it also talks a lot about my work. I'm also an author of 2 books called Curing Aging and Telomere Lengthening, and I am presently writing a 3rd book. I've studied my research and clinical studies, and everything has been in a lot of different areas in my past. These are kind of like the most important ones here. I've done a lot of studies in statistical theory, logic, probability, data analysis, and evolution, among many other things here. And all this education and knowledge has allowed me to become very good at what I call critical meta-analysis of scientific peer-reviewed studies, which means I'm really good at reading scientific peer-reviewed studies, even if they're in PubMed and they've been approved. They're not always accurate, but I'm really good at figuring out fact from fiction. I'm good at looking at statistical analysis, looking at data, and then looking at how the data was analyzed to really convince myself one way or another, is this paper fact or is it fiction? So this has been a really good tool for me to help stay on track and do the right things. It's not just aging. I've been involved in a lot of things that I've mentioned. I've invented a lot of things. I'm one of the inventors of a lot of things. Nobody invents anything all by themselves anymore, but I've played key roles in inventions of a lot of things. A lot of the big biotech breakthroughs like HGH, prochymosin. I've invented techniques like high throughput mutagenesis. Every time anybody does mutagenesis at a large scale, they're using my patents. TPA, tissue plasminogen activator, erythropoietin, on and on. If you go down, you'll see I led the research that discovered human telomerase. I also did the discovery of imeltistat, a telomerase inhibitor that's being presently in clinical studies for treating cancer. But there's a lot of other things I've invented too. One thing that I'm not really good at semantics. So I do say cure for aging, and I do get criticized for that a lot because a lot of people say it's not a disease, you can't cure it. Well, the other thing is I do believe aging is a disease, and I can't prove it, but I call it a disease, and I think that after we do cure it, everybody else will call it a disease too. I also tend to equate telomere lengthening with curing aging. I believe that lengthening telomeres is going to play a major role in curing the aging process. But there might be more, but I think that's going to be a major step. And so I refer to that all the time as curing aging. So sorry if my semantics get misunderstood sometimes. Also, when it comes to things like I'm calling these politics, again, I don't know if politics is the right word, but I'm a person who actually does not consider the FDA to be an obstacle. I don't think big pharma is an obstacle. I don't think society is an obstacle. But I'm going to, later on in this talk, will be the main purpose. I am going to talk about what the real obstacle is to getting aging cured. I'm a member of 2 associations, and I'm not a member of very many associations, but these 2 are really important to me: Coalition for Radical Life Extension and the Transhumanist Party. And that's because I'm not interested in just extending lifespan. I'm also interested in enhancing lifespan in every way imaginable. So these parties are very important to me, and I highly recommend everybody become members of these organizations. So I'm not going to spend a lot of time on the science. To learn everything you ever want to know, go to my website and go to this video called Everything About Telomeres. Now, unfortunately, it's 2 hours long, but what's really good is it begins with kind of a table of contents, just shown here, that tells you all the different subjects, and you can just go to the part where I actually talk about this. This is actually a presentation that I gave a continuing medical education conference about a year ago. So I went over in a lot of detail, but it is 2 hours long. And again, the place to go is sierrasciences.com, and the video is called Everything About Telomeres. Okay, in that thing, you'll hear that I have a lot of beliefs, and they're based on my critical meta-analysis of scientific peer-reviewed studies. But I'm going to give you right now just a quick overview of the things I believe, which would be contrary to what a lot of other people in my field believe. And I'm not going to debate them at all, because they could be right. But this is what I believe. And one is, I believe that aging is a recent evolutionary event. I believe if you look at the different branches on the trees, you can actually find that different branches age by different mechanisms, which tells me that aging pretty much evolved when a lot of these branches first got evolved. I think aging began somewhere between about 100 million to 1 billion years ago. Life on this planet did not have essentially an aging process before about 1 billion years ago. I also believe that, I said every branch has different methods of aging. I believe that there's multiple things going inside of all of us that cause aging. And I like to think of these things each as a stick of dynamite that's burning. But what's really important, I think, is what stick of dynamite has the shortest fuse, and that's the one that we have to target first. In humans, I believe that's telomeres. Now, I don't believe that mice have the same short telomeres. I believe that the sticks of dynamite with the shortest fuses are different in mice. And as Liz Parrish had mentioned, that mice are not necessarily a good model for studying human aging. I strongly believe that. I believe that really most of the work we have to do is in humans, with the exception of some maybe engineered mice that have been engineered to age like humans. And the stick of dynamite said that for humans, the kingpin of aging, or stick of dynamite with the shortest fuse, is, I think, our telomeres. And I believe it's not just us. I believe it's most primates, except for lemurs. Lemurs branched off from primates at a much earlier stage than other primates did. And then dogs, cats, horses, sheep, pig, and deer all appear to age like humans do by telomere shortening. But I've also come to the conclusion from my meta-analysis is that nothing else that we do to cure aging is ever going to work unless, in addition to that, we also stop telomere shortening. Telomere shortening is a clock that's ticking inside of our cells, and we have to stop that clock. It limits our lifespan to 125 years, no matter how good, healthy we are, how well we take care of ourselves, how our lifestyle is. It limits our lifespan to 125 years. There is no other theory of aging that is this absolute. This is why I believe that this is the first stick of dynamite we need to put out, and I call that, when we put that out, I call it cure for aging, even though I think we might have to do some more things after that. I also believe that telomerase, which is the enzyme that can lengthen telomeres, in humans, it's only found in our primordial germ cells. Those are the cells that produce our sperm and our eggs. And so it's not, a lot of people tell you it's produced in other cells, but I believe it's only found in our primordial germ cells. Also, there's rumors, contrary to popular belief, the telomerase does not cause cancer. It's just hearsay and rumors. It actually prevents cancer. And it also helps your body fight cancer to lengthen your telomeres. So that there is a scare going around, but there is not a single scientific publication that has shown that telomerase causes cancer. In fact, many have shown the exact opposite. And then this is another one. There's a lot of debate about why we age all to begin with. And I believe that we age as a way to shuffle our genes. When we shuffle our genes, it increases our chances of surviving a rapidly changing environment. I've got a strong background in evolution, but by knocking off the parents, as you see at the top of this slide, the young get a chance to breed, just thus shuffling the genes even more. Okay, so Sierra Sciences is in Reno, Nevada, and we're a discovery research company. All we do is basic discovery research, nothing else. And our only goal is to find a cure for aging. That's all we do. Telomerase, as I mentioned all before, this is a cartoon showing telomerase, is a kind of a factory. It looks like a factory in this cartoon. It binds to the end of the chromosome and it lengthens the telomeres. And that's what we need to do. Okay, so why don't we produce telomerase in any other cell of our body? That's been the big problem. We now know that it's because this is showing that we in every cell of our body, we have a gene for telomerase. Shown here is the telomerase gene. But it's shut off because there's a switch next to it, an on-off switch like a light switch, and it's shut off by a protein called a repressor. So one of the things that we're doing here at Sierra Sciences is that we are searching for drugs and natural products that would bind to that repressor and dislodge it, thus turning the telomerase gene on and lengthening our telomeres. So that's one approach that we're using. Another approach we're using is gene delivery, also known as gene therapy. The big circle is the human cell. I don't have slides as great as what Liz Parrish showed, but this is essentially the same thing. This is a human cell shown as a circle. The gene therapy is shown as a smaller, like soap bubble. Let's say to the right there, it contains the gene for human telomerase. And as we know, when soap bubbles fuse, the contents of one goes inside the other. When a gene gets inside the human cell, it then produces telomerase. And because we've engineered that telomerase gene to be powerful, it produces a lot of telomerase. So that is one method we're doing. This method's already set up. All we're trying to do is trying to get clinical studies done. All right, well, that's pretty much all I'm going to say about the science. If you want to know more, go back to my website and you find a lot of information, a lot more detail about that, especially that one video that I showed you. So why am I really here today? Or not here today, depends on your perspective. The reason is everything I've just said, I've been saying this for 30 years. And that's really frustrating. I've been saying it for 30 years and we still don't have aging cured. And I think that the problem, the obstacles that I said that I'm going to talk about later, is that funding and control of the science are always the obstacles. That is what's preventing the things getting done. It's never the science itself. And this isn't just for aging. I know people that have major cures for cancer, major cures for all kinds of diseases, and they can't get funding. You'll find announcements, people are making major announcements about big breakthroughs and stuff like that, but they really haven't made the breakthroughs. What they're trying to do is they're trying to attract investors, and nobody's succeeding. It's a really big problem. I founded Sierra Sciences in 1999. Okay, well, from 1998 to 2010, so a year before I actually started, I ended up raising $33 million over that, 12 years. And I got over 50 investors. I didn't find it difficult at all to find investors and find funding. That was really great. And what was also really great is that because of my background, I've done a lot of different things, as I mentioned. My scientific successes exceeded all the investors' expectations. So this was really great. So you would think we would celebrate. Well, the consequence of it was the investor said, let's sell the company. And this is really good. It makes the investors a lot of money, but that's not what I'm after. I'm not after the money. I want to do this research. I want to complete it. We had not cured aging when they were ready to sell the company. We had just made so many breakthroughs that the investors thought they could now get a great return on their investments because of the discoveries we made. And the problem with that is that that would have moved me out of the way, would have put somebody else taking over. And I've seen a lot of scientists, especially in the aging field, and a lot of them recently, whose whole careers have been destroyed by investors selling the company and taking the company out from under the person who founded the company. Fortunately, in my case, so what I want to say first is I've learned that investors invest in the dream. What they're trying to do is they're trying to make a quick return on their investment from the dream, but not necessarily the dream coming true. And this is what's happened over and over again. Most of these companies that I mentioned before, they started with a dream, they worked, investors turned the company into something else, sold the company became very successful, but the founders sure didn't think so. The founders were actually looking for work elsewhere and never actually succeeded at their original goal. I don't want to do that, and unfortunately, in my case, I got really lucky. A lot of fortunate events happened, and I was able to acquire 100% ownership of the company myself when the investors were trying to sell it to somebody else. So with that, I started a plan B. I decided investors was not the way to get something done. It was a way to get what they wanted to get done, but not what I wanted to get done. And that was cure aging. So I started a plan called the Go-to-Market Partnership Plan. In this case, I would retain 100% ownership of the company. I'm going to describe it in a little more detail in a minute. But I also started saying I will accept very large investments as long as I retain controlling ownership. The problem with when I had 50 investors, I only owned 17% of the company, which means I had no control over whether we sold the company or not. So the new business model that I'm pursuing, this is why I'm here. This is what I want to talk about. I call it a win-win-win situation. Okay, it wins for marketing partners. I want to get marketing partners to market discoveries that we have made, and we've made some very significant discoveries. And the marketing partners can keep 90% of the gross profits. So it's a win for them. It's also a win for Sierra Sciences because we keep 10% of gross profits to fund the research. And believe me, I think these products and therapies and treatments will be so significant that 10% of gross profits will actually end up being far more than we need to complete the research. The other winners are going to be finders. People listening to this conference, anybody who helps connect me with an individual or a company or somebody that wants to actually start marketing some of our discoveries, that finder, that person that brought us together, will get a 6% finder's fees. And I can tell you, I can put you in touch with plenty of people that have already done that and are really enjoying their 6% finder's fees. And then the general public wins because we'll actually get a cure for aging without all the obstacles. The obstacles, as I said, are the investors taking control of the company and not having enough funding. Now, I said I don't really want to bring in investors for Sierra Sciences research. I don't think Sierra Sciences is going to really making money. I think the go-to-market partners are going to make a lot of money. But investors can invest in the marketing partners. And this is something I really encourage. Nothing would make me happier than somebody investing in a company like BioViva so that BioViva can become a marketing partner of Sierra Sciences and hopefully start marketing a lot of what we have to offer. And what I want to do right now is I want to give a list, and I want to thank Liz Parrish for mentioning me a few times during her talk. She's the CEO of BioViva. Some of the go-to-market partnerships, or all of them practically, are right here. So nutraceutical telomerase inducers, we have a ton of them. We have pharmaceutical telomerase inducers, a ton of them. And these are good for skincare products, supplements, and even pets. So these things can be used. There's a lot of markets for these things. We also have telomerase gene therapy. We have all kinds of telomerase gene therapy, and we have it for humans and pets. We also have human growth hormone. As I had mentioned at the beginning, I'm one of the discoverers of human growth hormone. The discovery I made it so that human growth hormone could be made in bacteria in a form that doesn't cause an immune reaction. That was never patented. That was never published. It was kept a trade secret. I have it here. patents, if there were any patents and things, they've all expired now anyway. But I have the ability to, if somebody's interested in producing growth hormone, of licensing that technology to somebody so that they can produce human growth hormone. And the price that people are charging for human growth hormone is so astronomically more expensive than it costs them to make it, that there's a tremendous amount of profits that anybody could make by marketing this and competing with the other companies. Immortal pet cloning. We all know that there's companies now that you can send pet cells or pet tissue to, and they'll send you back a clone of your pet. Well, send us the cells first. We can actually immortalize them with our gene therapy techniques. And then you have a pet that will actually live longer and healthier than when, and we don't even know because nobody's done it yet. Telomere length measurement protocols. We've developed telomere length measurement protocols that blow all the other ones out of the water. Most of the reasons why telomere length measurement is not the best way to measure aging right now is because the techniques are so difficult and so inaccurate. But we've developed some techniques for measuring telomeres that are extremely accurate and far more informative than occur anywhere else. We also have the skill of total RNA sequencing. That means taking all the cells in the blood and sequencing all the RNA. And this is what true epigenetics is. Epigenetics is what genes are turned on and what genes are turned off. And when you sequence like 500,000 different RNAs inside of blood, you can actually see what genes are being turned on and off better than any other method available. And this isn't just true for the genes that are produced from our chromosomes, human genomic chromosomes, but also mitochondria and our microbiome. So we can actually look at everything, all RNAs. This technology exists here. We're just not a marketing company. We just do it for our research, but it is available if somebody wants to market it. As you know, my invention of the National Inventor of the Year Award in 1997 was actually for my cancer research. And so I understand cancer more than almost anybody else. I've got cancer diagnostics and cancer therapies that we have sitting here that if somebody wants to get involved in bringing these to the market, they're there for the terms that I discussed before. So our main goal is to develop a pharmaceutical telomerase inducer. That's my main goal. I want to see everybody take a pill and actually become young again when they swallow that pill. And I believe, and I've said this many times, I believe that we can have this pill in 1 to 3 years with the appropriate funding and with the appropriate control of the science. I've had the appropriate funding before, but I didn't have control of the science. It was too often that investors, some of them were good, but most of them would take over and direct us in other directions. And the money that we raised was actually spent on things that had nothing to do with what our major goal was. So I believe that we can have aging cured, and that's extrapolating from all our previous data. I actually have data showing that we can do this in 1 to 3 years. This is not just a guess. So again, funding and control of the science is the only obstacles. If we can solve this problem, aging will be cured. And again, let's all be there when this happens. And once again, the obstacle is funding and the control of the science. We can solve those problems. We can actually make all these things we've been talking about for many, many years come true. So thank you very much. My website's right there. You can find out anything. You can contact me through that website. And Nuno, this is a wonderful conference, and I look forward to listening to the rest of the speakers. Thank you.
Thank you, Bill, not just for this amazing talk, but also for being a visionary for more than 30 years, as you said it. And the soon we have the funding for leveraging the application of this technology, the sooner we can enjoy a long, healthy life. We're a little tight on schedule, but we're going to have it on the panel. So we're going to be able to have you addressing questions. I really want to thank you, and I want to tell everyone listening to us, especially for investors, just one second, sorry. Especially for investors, please take a look at the technology that Sierra Sciences is demonstrating and showing to work. Please take a deep look and take your own conclusions, because this may be what we need for a long, healthy life. Dear Bill, some final words you want to share?
No, I'll discuss it with later. I actually thought we weren't on right now.
Okay, sure.
Thanks.
Thank you so much for the great talk. Thank you.